Hello dear friends, welcome back again to another video from my YouTube channel. In the previous video, we talked about the transfer section of the young anther. There we have studied the four antheridial walls. The outermost wall, it is the epidermis, and the second outermost wall, it is the endothecium, and the third layer, it is the middle layer, and this fourth, as I told earlier, it is the very important layer in the anther, that is called as tapetum. This tapetum we are going to study in this particular video. Let us talk about the tapetum. Tapetum is the innermost layer of the anther. It is the innermost layer. It is the innermost layer. Now this tapetum, it is made up of conical shape or pyramid shape, compactly arranged cells. The cells of the tapetum, they are conical in their shape or they are pyramid in their shape, and they are also compactly arranged cells. These cells, as I told earlier, they are very special because they are all multinucleated cells. Let us take a closer look of the tapetum. Tapetum is a conical shape, compactly arranged, and they have got multinucleated. Multinucleated condition means if any cell is having more than one nucleus, then it is called as multinucleated condition. Look at the cell here. This cell, this particular cell, it has got three nucleus. That means it is trinucleated cell. Here we have got the four nucleuses. It is called as a tetranucleated cell. Here it has got, this tapetal cell has got only two nucleuses, then it is called as binucleated cell. Simply all these tapetal cells, they are multinucleated cell, and this multinucleated condition in the tapetum, it is due to endomitosis process. Now, tapetum is the most important layer in the anther. Why it is most important? After studying the functions of the tapetum, you will agree the importance, or you will agree it is the most important layer for a anther. The first function of the tapetum consists of various nourishing material in them. These cells are all filled with nourishing material, abundant amount of nutritive material, nutritive substance is present, and this nutritive substance, it is present in the form of protoplasmic content. Hence, tapetum is a nourishing tissue. Tapetum is the nourishing tissue. Now the question arises, if it is a nourishing tissue, this is giving nourishment to whom? It is giving nourishment to the pollen grains, to the pollens. Where these pollens are located? Pollens are located here, and this is the tapetal layer. This is the tapetal layer made up of tapetal cells. Inside this, there are pollen grains are getting synthesized. During the synthesis, during the development, during the nourishment, during the maturity of the pollen grains, all these pollen grains are getting nourished by the tapetal cells. The first function of the tapetum is to nourish the pollen grains. Then the tapetum secretes an enzyme. Tapetum secretes enzyme. Tapetum secretes enzyme, and the name of the enzyme is Calase enzyme. Calase enzyme. We'll come to know about this calase enzyme little later in our future videos, because this calase enzyme is used to dissolve the callose material which is present in the microspore tetrad, that we're going to discuss while discussing the microsporogenesis process. Now, the second function of the tapetum is to secrete an enzyme, calase. Tapetum also secrete one more very important substance. Tapetum secretes another very important substance, that is sporopollenin. Sporopollenin. We have discussed this sporopollenin while we are discussing the structure of the pollen grain. Sporopollen is the highly resistant material that forms the exine to form exine in pollens. The outermost layer of the pollen grain is called as exine, and this exine is made up of a substance called as sporopollenin, which is a non-degradable substance. No enzyme can degrade the sporopollenin, no acid can degrade the sporopollenin, no base have got any activity on the sporopollenin. It is highly resistant material. This highly resistant material, sporopollenin, is getting secreted by the tapetum. Now, the fourth function, tapetum, it secretes the pollen kit, or it produces the pollen kit. Tapetum produces pollen kit. Pollen kit. What is this pollen kit? Initially, if this is the tapetal cell, which is a conical shaped cell, multinucleated cell with protoplasmic content. This is the tapetal cell. Tapetal cell initially produces certain bodies, certain structures. These structures are called as Ubish bodies. Ubish body or Ubish bodies they are. Tapetal cell produces the Ubish bodies. These Ubish bodies, they produces the pollen kit. Now, what is this pollen kit? Pollen kit is actually a yellowish substance. Yellowish material around the pollen grain, around the pollens. Pollen grains are generally yellow in their color, and that yellow color is due to the pollen kit, and this pollen kit is produced by the Ubish body. Ubish bodies are getting synthesized by the tapetal cells. Now, why the pollen grains are surrounded by a yellow color material? To attract the insects for the pollination. Yellow color is a bright color, and this bright color attracts the insects for pollination. That pollen kit is produced by the tapetal cells. They are the four very important functions of the tapetal cells. In most of the competitive exams, based on this functions of the tapetum, questions were asked. Now, coming to this structure, this portion of the anther. This portion of the anther, it is called as the pollen sac. It is the pollen sac. Pollen sac. In this pollen sac, so many cells are present. Many cells are present. All these cells, they are the MMC, microspore mother cells they are, or they are also called as PMC, pollen mother cells. Here we have group of pollen mother cells, or we have simply the group of microspore mother cells. Collectively, these pollen mother cells or microspore mother cells, they are called as a sporogenous tissue. Inside the pollen sac, there is a sporogenous tissue or microspore mother cell, or the pollen mother cell. At the center of this transfer section of the anther, here we have a sterile tissue. This sterile tissue is nothing but the connective tissue. Why it is called a sterile tissue? Because it is not playing any role in the reproductive process. It is the connective tissue that connects anther with the filament. That is the main job of the connective. In our next video, we'll discuss about how the pollen grains are produced from pollen mother cell inside the pollen sac. That process is called as microsporogenesis. That we'll discuss in our next video. Thank you.